Amplicon sequencing of ribosomal subsequences can characterize microbial community composition with the potential to extrapolate genomic functions. Methodology to predict bacterial community function from amplicon sequencing has been developed, but no fungal equivalent currently exists. The FUNTFITS pipeline aims to resolve this issue by pairing annotated genomic data from the Joint Genome Institute with fungal ITS amplicon sequencing data to generate estimated EC gene counts for diverse fungal communities. However, some ECs are consistently over or under predicted across samples by the existing FUNTFIT pipeline, as shown in the upper left results plot. To improve the prediction pipeline, we aim to uncover biologically meaningful per EC and per genus weights that reduced error to incorporate them into the pipeline.
We first attempted to use genome completeness as a measure of confidence. Within the fungal community, there has been a push to sequence more genomes, as many genomes contain gaps in their sequencing and are incomplete. This part of our project explores the hypothesis that taxa with low sequencing completeness have decreased EC count concordance between FUN2FIT's predictions and the metagenome. We evaluate genome completeness using BUSCO completeness scores. We expect taxa with high BUSCO completion scores to have slopes closer to 1 or high concordance with the metagenome, and taxa with low BUSCO completion scores to have slopes closer to 0 or low concordance with the metagenome. The dot plot shows the slope for each genus along with the dots coloring showing the mean completeness score for all taxa within the genus. Green indicates that the mean genome completeness is over 90%, while pink means that the mean completeness is below 90%. This plot displays a lack of relationship between slopes closer to 1 and higher genome completeness. The scatter plots to the right of the poster highlight the variance in concordance between metagenome EC counts and FUN2FITS counts by genus. The top plot showcases the best scoring genus, Incertiate genus, which are the genomes not determined at the genus level. The plot on the bottom is Penicillium, one of the lowest scoring genera, with many metagenome EC counts having no FUN2FITS predicted counts. In conclusion, these results show a lack of relationship between genome completeness score and concordance between FUN2FITS and metagenome EC counts.
In addition to weighting genomes by their completeness, we hypothesized that EC phylogenetic signals can be used as a measure of confidence in our predictions. Since the JGI tree is used to place species without sequenced genomes, and EC counts are predicted by a species' nearest neighbors on the tree, the accuracy of this tree influences FUN2FITS' prediction accuracy. If the tree is an accurate predictor, we expect that the mean error will correlate inversely with the phylogenetic signal and can be used to weight the prediction confidence. We used Blomberg's K to measure the phylogenetic signal of normalized and unnormalized EC counts across the JGI tree. High phylogenetic signal means that the relatedness between species correlates well with EC counts and will lead to a higher K. A p-value can also be calculated by randomly shuffling the EC counts among the tips and recalculating K for each permutation. The two graphs right here compare Blomberg's K and the p-value for permutations of Blomberg's K for normalized EC counts on the x-axis and mean pipeline prediction error on the y-axis. There is no correlation between K and the mean error and p-value and the mean error, meaning that EC count prediction accuracy of the pipeline does not depend on the phylogenetic tree. The lack of correlation between phylogenetic signal metrics and prediction accuracy is surprising, given that FUN2FITS heavily relies on the phylogenetic tree to predict EC counts. However, it shows us that the error in the pipeline is likely not due to the phylogenetic tree and has some other root cause.
Last, we attempted a naive hill climbing approach for learning genus weights with methodology displayed on the left side of the poster slide. We know that many of the poorly predicted EC numbers are consistently over or under predicted. If this is due to genus bias in ITS sequencing, we expect that stochastic learning of genus weights would result in a consistent signal of prediction biases. While the hill climbing method showed consistent decrease in error as seen in the middle plot, the final weights generated by different random seeds diverged further with more rounds of hill climbing. If the learned weights were consistent, we would expect to see distances between differently seeded weights converge towards 0. In the right plot, leave one out cross-validation of weights showed an average error decrease of 2.25%, and while most samples showed a decrease, 2 samples showed a strong increase in error. These samples contain genera that were not well represented across samples and demonstrate additional limitations of this method. This method shows initial promise for decreasing error, but more work must be done to uncover why weights diverge in a non-deterministic manner and what the biological significance is of these weights.
Overall, we analyzed 3 different methods for improving the accuracy of the Fun2Fits pipeline. Analysis of error contribution of EC phylogenetic signal and genome completeness did not explain the high mean error observed. Leave one out cross-validation of the machine learning method showed 2.25% mean decrease in relative error. Our findings suggest machine learning can be utilized to improve pipeline performance, but more work must be done to uncover the biological significance of prediction errors.
Thank you.
Thank you.
Thank you.